reproductive system is divided into the male reproductive system and the female reproductive system. This lecture will cover the male reproductive system. For all systems, these items are the same. Both males and females have gonads. These are the organs that produce both the gametes and the hormones. They also have ducts that are going to move those gametes around through the body, glands that help with reproduction. These are the accessory glands, and then the anatomical perineal structures. Most of the time, these are called the external genitalia. Both male and female reproduction are different. The female makes 1 gamete per month on average, sometimes 2. We'll talk about twins a bit later. And not only does the female produce the single gamete or 2 gametes per month, the female also retains and nurtures that fertilized gamete, that oocyte that becomes fertilized. And once that occurs, it is then called a zygote. Males, on the other hand, produce about 500 million sperm per day, very large quantities of gametes. And if we go back and think about why a male would produce this number of sperm per day, remember, humans are different, and they're the most unnatural of species. Traditionally, males are not bound by any cultural or religious ties, and their job is to go around and fertilize as many females as possible, because when it comes down to it, our job on this planet is to get our genes out in any way that we can. The male reproductive system has testes, also known as male gonads, and they secrete the male sex hormones, which are known as androgens. And they also produce the gamete, which is known as sperm or spermatozoa. Emission is the term that is used to describe the mature sperm moving along the duct system. You may have heard the term nocturnal emissions, maybe when you took a psychology class. Well, that term is actually not correct, because emission does not mean that the sperm leaves the body. Emission just means that the sperm is moving through the body. Semen is sperm mixed with fluids so that it can be emitted through the body, and ejaculation is a term for when sperm is expelled from the body. Once sperm is made, it has a path that it has to follow, going from the testes that you can see right here to the epididymis, to the ductus deferens, or the vas deferens, you can call it either. You follow that, it then goes to the ejaculatory duct that you can see right here, and then it hits the urethra. Accessory organs are also involved with sperm, and they secrete fluids into the ejaculatory duct and the urethra. This includes the seminal vesicles, which are right here, the prostate gland, which is right here, and the bulbal urethral glands, which are also called Cowper's glands. Along the path for sperm or other organs that are involved, we have the external genitalia, to include the scrotum. It encloses the testes, and then the penis. That contains the distal portion of the urethra. The testes, testis is singular. The testes are egg-shaped. They're about 5 centimeters long, 3 centimeters wide. They hang in the scrotum. The scrotum is a fleshy pouch. It hangs inferior to the perineum, anterior to the anus, and posterior to the base of the penis. When we look at the formation of testes in the fetus, it's rather interesting where the testes begin and then where they end. So if you look at the image, you see that we're looking at a 2-month, a 3-month, and then a fetus that is at birth. So what happens to the testes? Well, the first thing that you notice is that in the fetus, there's this structure called the gubernaculum testis. This is a cord that's made of a connective tissue, and it goes from each testis to a pocket in the peritoneum. And what the job is to lock the testes in place as the fetus grows. So it's not that the testes move, it's that the fetus grows with the testes being locked into this particular position. So when you look at what happens during the 7th month, we know that a fetus will grow rapidly. Sometimes it will double in size. And hormones that have been produced by the testes will allow for the testes, gubernaculum testes, to then contract. Up until the 7th month, there had been no movement. At that time, each one of the testes will move through the abdominal wall. You can see right here through the abdominal musculature. And along with that movement, there are pockets in the peritoneal cavity. Now, the testes also have accessory structures that accompany it during its descent, and that forms the spermatic cord. So you can actually see the spermatic cord here and here. You also have the ductus deferens that's formed, and you have the blood vessels, nerves, and the lymphatic vessels. All of that is within the spermatic cord. All right, so here's an adult spermatic cord. You can see a deep view on this side and a more superficial view over here. So you see the circling of the spermatic cord itself, and it goes between the abdominal pelvic cavity and the testes. So you can see that this is an adult male where the testes have fully descended from the abdomen. When an infant is born, a male infant is born, those testes are going to be held very close up to the body, and some of them may not have descended fully, either one or both. So the clinician may give it a few days to see if the testes descends on its own, on their own, and if they don't, then simple surgery is done to allow for the testes to drop. So the spermatic cord has fascia, it has muscle, and you can see in this deep picture, you can see the vas deferens, blood vessels, nerves, and lymphatic vessels. So the spermatic cord passes through the inguinal canal. So I'm circling that inguinal canal right here. So it is a hole or a passageway through the muscles of the abdomen. So as the testes descend into the scrotum, it has to make its way through this hole. Males will at times get what are called inguinal hernias, and that is when you have tissues that protrude through this opening in the abdominal wall. And because you have the spermatic cord that has made this passageway, it causes a weak point in the abdominal wall. So if a person is overweight or they lift heavy things often, or they're doing it incorrectly, they can get an inguinal hernia. The scrotum itself has 2 chambers, and each testis lies in a separate chamber. Surrounding the testes is the dartus muscle. So this is the dartus muscle right here. It's a layer of smooth muscle, and it causes the characteristic wrinkling of the scrotum. The cremaster muscle is deep to the dartus muscle, and that is what I am showing you right here. That is the dartus muscle. This allows for the scrotum to pull the testes closer to the body, and this has to do with temperature regulation. Sperm are not functional when they're hot. So the testes evolved so that the sperm can be kept further away from the body, and it lowers the body temperature by about 2 degrees Fahrenheit. So if you have men who wear boxer shorts or ride their bicycle a lot or stay in a hot tub, it can actually lower sperm count because sperm prefer to be at a lower temperature. Now, if we look at the structure of the testes, this is the frontal section of a testes, right? So it has been transected. And if you look at this picture, you see the cremaster muscle, just to give you a point of reference. This is the scrotum itself. The dartus muscle is located right here. You can see the testes, and remember there's the structure that sits on top, and that is the epididymis, right? And this is the opening to the vas deferens or the ductus deferens. All right, so going from the inside, or excuse me, from the outside in, here is the tunica albuginea. This is connective tissue, has lots of collagen, and it's continuous with all of the fibers that surround the epididymis up here. So this is tunica albuginea here. Also, you have portions that extend into each testis, and each one of those extensions will converge, and they support the blood vessels and the lymphatic vessels and the afferent ductules. Now, another thing that you saw within the testes was the seminiferous tubules. So if you think about the seminiferous tubules, if you straighten them all out and you think about them like a garden hose. So this cartoon is a cross section of a seminiferous tubule, right? So I'm now highlighting the wall of the tube, all right? So this is the wall of the tube that I'm marking up in the green. This wall is so incredibly important, and you can see the same thing over here in the actual photomicrograph. You can see that this is the seminiferous tubule, cross-section of the seminiferous tubule, and this area is the wall. It encircles the lumen, which is the space. So again, think about it like it's a garden hose. But I want you to think about the wall of the seminiferous tubule, because as you can see, it's incredibly important. When you look at the cartoon, you can see that there's all sorts of meiosis going on in this wall of the seminiferous tubule. So we have septa that divide the testes itself into these different lobules, and inside the lobules you can see all of these coiled seminiferous tubules. There's about 800 of them. This is where sperm is produced within the seminiferous tubules. Each one's about 80 centimeters long. Each testis has about a half mile of seminiferous tubules. Each one of the lobules will have the tubule that goes up to the rete testes, right? So this is the rete testes. Oops, my highlighting got a little crazy there. This is the rete testes, and the rete testes will then feed into the epididymis. So we're going back to the wall of the seminiferous tubules. So there are different types of cells, either surrounding the tubules or within the tubules themselves. So the first one I want you to notice is a type of cell that surrounds the wall of this, or surrounds the seminiferous tubules. Interstitial cells produce androgens, right? They produce testosterone. This is the most important androgen, right? So you have interstitial cells here, here, here. So clearly they are related to what is occurring within the seminiferous tubules. The process of spermatogenesis is the process of sperm production. So in this picture, you're looking at a pie-shaped wedge, a piece of the wall of the seminiferous tubule. All right, so if we look outside the wall, you can see capillaries, interstitial cells, all of those are outside the seminiferous tubules themselves. Now, as we move in, you can see that there is this dotted line, right? This is called the blood-testis barrier. On the lower portion, or on the lower side of that line, is this area that I am highlighting. And in this area, in this outermost cell layer, this is where spermatogenesis is going to begin. Each one of these cells that I'm circling, so there's 6 of them that are being shown here, those are called spermatogonium. Spermatogonium is the term for stem cells, sperm stem cells. Spermatogonium are sperm stem cells. So if we look at what happens to spermatogonium in this picture, here we have a spermatogonium, and it is a stem cell. And so if you remember what we've talked about at stem cells, when stem cells go through mitosis, they essentially have 2 jobs with the daughter cells that are produced. One of the daughter cells is going to remain a stem cell. That's what this one's doing. The other daughter cell is going to go and do the job of whatever is requested. And so this stem cell is eventually going to go through meiosis, and it's going to become 4 sperm, right? So just very quickly, even though we're in male reproduction, I want you to see that both forms of gamete formation start with stem cells. Here we're starting with the spermatogonium. It then moves, starts to go through the process after dividing via mitosis to produce the daughter cells. What that daughter cell that goes on to be part of the process, it is called a primary spermatocyte, right? That primary spermatocyte is going to divide due to meiosis 1, and it's going to produce 2 daughter cells, and those are secondary spermatocytes. Those 2 daughter cells are then going to divide each one, and they're going to produce 2 daughter cells. And then those 2 daughter cells will mature. So we go from a primary spermatocyte to a secondary spermatocyte to spermatids, and then to sperm. If you look at it compared to a female, you can see that again, stem cell goes through mitosis. The daughter cell that it's going to go through the process is called a primary oocyte. That primary oocyte divides in meiosis 1 and produces 2 secondary oocytes. These secondary oocytes, only one of them is going to go through meiosis 2. And when it goes through meiosis 2, only one of them is going to eventually become an ovum. So what I want you to think about is if each one of these primaries, whether it is a primary spermatocyte or a primary oocyte. Let's just talk a crazy number. Let's just say, how much DNA is on the inside of this primary spermatocyte? And I'm going to give you a crazy number. I'm going to say 12 pounds, right? It's crazy. But when I give you the extremes, I think you'll see why. So if the DNA, all of those chromosomes on the inside weigh 12 pounds, how many pounds of DNA do you find in the secondary spermatocytes? Well, it divided everything in half, right? So each one of these has 6. All right. Now, when we get to dividing again, each one of these has 3, right? 3 here, 3 here, 3 here, and 3 here. Now, I want you to look at the DNA between the spermatid and the sperm. Did the DNA change? It did not. So the DNA here is 3, 3, 3, and 3. All right. Now let's go over here and look at the oogonium. Same amount of DNA. So the primary oocyte has 12 pounds of DNA. The first division, meiotic division, what do we have? Same thing, 6 and 6. But we produce this polar body for a crazy reason, and I'll get to it in just a minute. So the secondary oocyte divides, and what happens to the DNA? We have 3 here and 3 pounds here. And then from the ootid to the ovum, the DNA doesn't change. 3, right? Started with 12. Now we have 3, but we have 3 for each one. So I want you to think about this. If this sperm fertilized this egg, or this oocyte, you'd have 6 pounds of DNA. The first thing that's going to happen is you're then going to have DNA synthesis. So when you do that, you actually double the amount because you're synthesizing DNA. So when you do that, you're back up at 12. And what you have is a combination of the male and female, but you have it in the right order and the correct amount from each parent. That's if we have homeostasis and all of this occurred correctly. All right, so no, you don't have 12 pounds of DNA in a cell, but I'm trying to make the point that when sperm are made, this is an easy division. You have 4 products, each with 3 pounds of DNA, so 4 times 3 is 12. Well, you only make 1 egg or 1 oocyte, so we can't go through the same process. We have to get rid of some DNA in order to make sure that the female product only has 3 pounds. So the way we do that is to make what are called polar bodies. And that gets rid of the excess DNA. I'll talk more about that when we get to females, but I just wanted you to see why we go through this process of spermatogenesis and oogenesis. Spermatogenesis is a little easier to understand because you have 4 products, each has equal amounts of DNA in them, and you basically are mimicking the amount of DNA that's in the primary spermatocyte. All right, so in order to make sperm, you have the stem cell, the spermatogonium. It divides by mitosis, produces 2 daughter cells. One remains spermatogonium, and the other one differentiates into what's called a primary spermatocyte. That primary spermatocyte begins meiosis. With the first meiotic division, you form secondary spermatocytes. Those secondary spermatocytes will then, through the second meiotic division, will become spermatids, and these are immature gametes. Those spermatids differentiate into spermatozoa, and those spermatozoa they're in the wall of the seminiferous tube. Remember, all of this is occurring within the wall of the seminiferous tube. The spermatozoa will actually lose contact with the wall, and they enter the lumen, right? They enter the fluid within the lumen. Okay, so there's something called Mullerian ducts, and so both males and females have Mullerian ducts. So as you can see in this picture, this is male and female embryos, and you can see the differentiation here into male and then here into female. But initially, both males and females had both Mullerian ducts and Wolffian ducts. So let me talk about the Mullerian ducts first. Mullerian ducts, if they're maintained, will become the uterus and the uterine tubes and the cervix and the most superficial part of the vagina in females. Adult or males do not have Mullerian ducts. So males produce something called Mullerian inhibiting factor, right? Mullerian inhibiting factor allows for the Mullerian ducts to be inhibited. So that is what a male who is in homeostasis has. This Mullerian inhibiting factor, and it allows for the ducts to be reduced in the male, so they don't have the structures that are associated with a fully functioning Mullerian duct, because those Mullerian ducts will become the uterine tubes and uterus in females. But if a male doesn't have enough Mullerian inhibiting factor, he will retain those ducts, and the testes may not descend into the scrotum. Now, there are numerous reasons why testes don't descend into the scrotum, but this is one of them. All right, Wolffian ducts, on the other hand, these are the structures that lead to the male internal genitalia. Again, you find them in both in males and females that are fetal, early in their fetal life, but they will regress in females, and you can see that degenerating Wolffian duct right here. But in males, you can see that they are maintained, and what they become is the vas deferens, the epididymis, and the seminal vesicles. So when you hear people say there's a time in your life when everyone's female, this is what the discussion is about, because Mullerian ducts actually become the uterus and fallopian tubes. Okay, so it's one thing to produce sperm, but it's something totally different to produce sperm that can fertilize an oocyte. So when the testes produce the sperm, when these little guys right here enter the lumen and they travel through the lumen, they're physically mature, but they cannot fertilize an oocyte yet. The rest of the male reproductive system allows for something that's called functional maturation. So those sperm are going to be released from sustenacular cells. So here are the sustenacular cells that you see within the wall of the seminiferous tube. So they release from those cells, they go into the lumen, but they are considered to be functionally immature at this point. So keep that thought about functionally immature sperm. So the sperm are loosed inside of these seminiferous tubules, and they're making their way toward the reed testes that you can see right here. All right, but let's get up to the epididymis. So this is the epididymis. This structure is the epididymis. It's about 7 meters long if uncoiled. It has the jobs of fluid monitoring and adjustment. The fluids that are produced by the seminiferous tubules are going to be adjusted and monitored by the epididymis. Damaged sperm are going to be recycled here, and also storage and protection of the sperm. The epididymis also allows for the functional maturation of the sperm to occur. So when sperm leave the epididymis, they're mature, but they're not swimming. They're just moving. So to become active swimmers and become totally functional, they have to undergo something that's called capacitation. So the first thing that happens in capacitation is the sperm become swimmers when they're mixed with the secretions from the seminal vesicles. So if you go think back, I showed you 3 accessory structures. One of them was seminal vesicles. And then they totally become able to fertilize and oocyte when they are exposed to the female reproductive tract. So an ejaculated sperm, if not exposed to the different substances in the female reproductive tract, they cannot fertilize and oocyte, right? So there's 2 steps to this capacitation. All right, now that we're out of the epididymis, let's think about the vas deferens. So this structure, these structures right here, are the vas deferens or the ductus deferens. So inside the vas deferens or ductus deferens, sperm can be stored for several months, and they are in a state of suspended animation where they're not utilizing nutrients. The vas deferens are about 45 centimeters long, and they start at the tail of the epididymis, and they continue on and go through the inguinal canal. So they started way over here at the testes, come back up, here's the testes, come back up, start right here, or I should say start at the epididymis, and come all the way up. And as you can see, they go through the inguinal canal and they become part of the spermatic cord. All right, so I pointed out the ejaculatory duct earlier, but here it is, and it's a very short passageway, about 2 centimeters, and it's at the junction of the seminal vesicle duct, which is right here, and the ampulla of the ductus deferens, right? It goes through the prostate gland, so this structure is the prostate gland. It goes through the prostate gland, and it empties into the urethra, right? The male urethra has 2 jobs. It has a urinary system job and it has reproductive system job. It's 18 to 20 centimeters in length, and it goes from the urinary bladder, as you can see, the urinary bladder is right here, all the way to the tip of the penis. There are 3 regions: prostatic, membranous, and spongy. Seminal fluid is a mixture of secretions from multiple glands, and each one of those secretions has very distinctive characteristics. So these glands include the seminal vesicles, the prostate gland, and the bulbourethral glands. So the glands are there to activate sperm, feed the sperm, propel the sperm because of peristaltic contractions, and they produce buffers. So the seminal vesicle is the first one. It's a tubular gland, it's about 5 centimeters long. It has short branches from the body. It's very active and it produces about 60% of semen. The seminal vesicles secretions are alkaline. That's to neutralize the acids both in the prostate gland and in the vagina. So the vagina is a highly acidic environment. It's actually called the hostile environment of the vagina. The acidity is incredibly important to keep down bacteria, but it is a hostile place for semen. So having an alkaline solution helps to buffer that. Also, because of the seminal vesicles, that first step in capacitation, where the flagella begin to beat and the sperm become swimmers. The secretions are discharged into the ejaculatory duct, as I showed you earlier. And this happens when peristaltic contractions are underway. And all of this is controlled by the sympathetic nervous system. The prostate gland is a small muscular organ, about 4 centimeters in diameter, and it encircles the proximal portion of the urethra, right underneath the urinary bladder. This solution is slightly acidic. It forms 20 to 30% of semen volume. It has antibiotics in it, and it is ejected into the prostatic urethra, right into the urethra itself. It's not going into a duct. And this occurs because there is peristalsis in the prostate gland wall. The tiny little bulbourethral glands, here they are, right, one right here, is found in the urogenital diaphragm. They're about 10 millimeters in diameter. They're found at the base of the penis. They produce a thick alkaline mucus to help neutralize acids again. So again, it's to help with the pH. It lubricates the solution within the penis, and it empties into the lumen of the urethra. So it just dumps in at this point.